A whole skeletal muscle is made of many groups or bundles called fascicles. Each fascicle is a bundle of individual muscle cells. A single muscle cell is also called a muscle fiber because it runs the entire length of a muscle. Voluntary contraction of skeletal muscle originates in the brain and is communicated to the muscle by motor neurons, each of which is connected to many muscle fibers. Let's look at the relationship between a motor neuron and one muscle fiber. Electrical impulses or action potentials from the neuron are passed to the muscle fiber at a specialized structure called the neuromuscular junction. In the enlarged area, note that the membranes of the motor neuron and the muscle fiber are very close, but they don't actually touch. The membrane surrounding the muscle fiber is called the sarcolemma. The portion of the sarcolemma just under the motor neuron is called the motor end plate. The neuromuscular junction is an example of a synapse, and two of its main structures are derived from that word. First, the enlarged end of the motor neuron is called the synaptic terminal. And second, the space between the synaptic terminal and the motor end plate is called the synaptic cleft. To initiate contraction, the electrical signal from the neuron must be transmitted to the muscle fiber at the motor end plate, exciting it. Let's zoom in even more closely to see that signal mechanism. Inside the synaptic terminal are small membranous sacs called vesicles. These vesicles are packed with a chemical called acetylcholine. Acetylcholine is often abbreviated ACH. Embedded within the motor end plate are acetylcholine receptors. These proteins bind to or receive acetylcholine when acetylcholine is present in the synaptic cleft. Within the synaptic cleft is an enzyme called acetylcholinesterase, abbreviated ACHE. This enzyme is always present within the cleft and breaks down acetylcholine that is released from the vesicles into the synaptic cleft. When a neuron is stimulated, an electrical impulse called an action potential arrives at the synaptic terminal. The arrival of the action potential causes the vesicles to fuse with the plasma membrane. As they fuse, the vesicles open up the synaptic cleft, releasing acetylcholine through the process of exocytosis. Acetylcholine binds to the receptors on the motor end plate. This binding increases the permeability of the motor end plate to sodium ions. Sodium ions rush into the sarcoplasm down their concentration gradient. This generates an action potential in the sarcolemma. The muscle action potential then travels along the sarcolemma, and this is what causes the muscle fibers to contract. Excitation of the muscle fiber ends as acetylcholine is removed from the cleft by acetylcholinesterase. Some of the breakdown products of acetylcholine are then reused by the neuron to make more acetylcholine and refill vesicles. With acetylcholine cleared from the synaptic cleft and synaptic vesicles refilled, the neuromuscular junction has now returned to its resting state. Let's review the process that takes place at a neuromuscular junction. Step 1. The neuron and the muscle are in a resting state. Step 2. An action potential arrives at the synaptic terminal. Step 3. The release of acetylcholine into the synaptic cleft occurs when vesicles fuse with the membrane. Step 4. Acetylcholine binds to receptors on the motor end plate, increasing the permeability of sodium. Step 5. An action potential is generated and propagates along the sarcolemma, ultimately causing the muscle fiber to contract. During this excitation phase, acetylcholinesterase breaks down acetylcholine in the cleft, which terminates the transmission from the neuron. As the action potential sweeps across the sarcolemma, the neuromuscular junction returns to the resting state, awaiting the next action potential.